right, good afternoon, everyone. So today we are going to finish off our Chapter 4 slides. I'm not quite sure how long it's going to take, but I think we should come out right at the end. So we'll start Chapter 5, I believe it is, next week. So we'll move on to our next topic. And just a reminder, because I keep forgetting, Monday is Thanksgiving, so there's no classes. So please don't come in, and I'm going to try and remember not to come in as well, because I have definitely shown up on campus when I'm not supposed to. So finishing off, we had spent the very, very end of last class talking about the difference between additive and subtractive color mixing, mostly to draw your attention to the fact that what we think of in terms of color mixing is actually subtractive color mixing. That's like mixing paints together. And as you add to it, you actually end up getting darker and darker colors. But when we're adding with light, we are indeed adding extra wavelengths to what we're looking at, and we get lighter and lighter in time. And I had pointed out that there's a handy-dandy little YouTube video about color mixing and additive versus subtractive color mixing, if these concepts weren't quite clicking for you. And I find that one helps quite a bit for me. But the reason why we talked about additive versus subtractive color mixing is because we're going to be talking about some different theories of how we perceive color. And these theories are based on additive color mixing. So if we remember that white light, the light that's coming from things that are mostly white, is going to be white because it has all of the different wavelengths that we can see. So everywhere from that 400 nanometers up to the 700 nanometers, more or less. So the more wavelengths that we have in that range, the closer we're going to see to pure white light. So how do we get to pure white light and all of the different colors in between using only our cones, those sensory receptors that we have that allow us to perceive color? And so there's a bunch of different theories. And you're going to find that the first two that we start with do a good job explaining part of our color vision, but on their own, they can't give you the whole picture. So instead, we're going to use them as building blocks to get to what we think of nowadays. And I do give the warning that these theories, we kind of keep it, again, sort of high level. We don't go into lots of details because we could spend a lot of time down this rabbit hole. We kind of gloss over it. And if you have more questions, there's a course on sensation and perception where you get to do this for the entire semester. And we talk about it more there. But first theory is the trichromatic theory. And if we think about it, tri meaning three, and chromatic meaning to do with three colors. So this theory focuses in on the fact that the human eye has three different types of color-sensitive receptors. So what that means is there are three different kinds of cones. And they're going to be sensitive to different wavelengths or different colors of light. And so this theory is pretty old. This was proposed in the mid-1800s. You'll sometimes see this called the Young-Helmholtz theory, and that's because it was proposed by two theorists, Young and Helmholtz. But as per usual, I don't care too much about the names. I like the trichromatic theory because that gets to the heart of three different color-sensitive cones that we have. And so those three different sensitivities are going to be specific to the wavelengths of red, green, and blue, which we had momentarily noted last time when looking at our additive color mixing, that red and green and blue mixed together should give us white. And the reason why is because red is at one end of our visible spectrum, and blue is at the other end of our visible spectrum, and green is somewhere in the middle. So by having all three, we get a decent overlap of different wavelengths. And so our colors that we actually perceive are created by an additive mixing process. So we're going to look at the comparative amount of firing for those blue cones versus the green cones versus the red cones. And so this diagram here is meant to show sort of that curve being the sensitivity of those cones to different frequencies of light or different wavelengths of light. So where the peak is, that's where they are most sensitive. So we can see that our blue colored cones have that peak right around, where is that, 440 nanometers? That would be approximately where they fire the most. And then as we get away from that, we would see less intensive firing, so a reduced firing rate as we deviate from what they're sensitive to. So if we move a little bit higher in wavelength, or a little bit lower in wavelength, we'll see slightly different patterns of responding, usually just slower responding. And so for our brain, if we were looking at, let's go into, say, the 480 nanometer wavelength there. So that's going to have a little bit of firing from from all three. So equal firing of blue and green, and then even less firing from red. And so the idea here is that our brain would be getting signals from some of our blue sensitive cones, some of the green sensitive cones, and some of the red sensitive cones, and it would say, okay, well, we have this much firing for these kind, this much firing for those kind. What does that mean? Where do we see that overlap? And so it would be like, okay, well, that's about a wavelength of 480 nanometers. So what we're seeing when we put all of this together is something in that sort of light blue turquoisey range. And so we could use all sorts of different combinations of those three firing in order to figure out what frequency it is that we're seeing. Now you might notice that it is a little bit difficult to tell sort of in that yellow range, because the red and the green are very, very close in their peaks. And they overlap a ton where yellow is. So from this theory, it actually has a hard time explaining our ability to perceive sort of different shades of yellow. But it does a pretty good job on everything else. And considering how old this theory is, not too bad. And if we wanted to look at what this theory is really good at explaining, it does a great job explaining color blindness. So for most people who are colorblind, they tend to be dichromats. So if we had that trichromatic theory, was looking at three kinds of cones, then dichromats only have two kinds of cones. So they're missing either the red, the green, or the blue sensitive cones. And that seems to be consistent with most types of color blindness. There are special cases where some people are monochromats, where they only have one kind of cone, and they have a lot of trouble with color vision. But most people that we see are dichromats, and we tend to see people who have trouble telling red and green apart, but there are other kinds of color blindness that are out there. So there's three big kinds of dichromatism, so insensitivities. And I include this neat little website at the bottom. This is just for your interest. I've done this class before and have had students who are colorblind or people who have members of their family who are colorblind. A couple of my cousins are colorblind, and it's one of those sex-linked traits that's more common in males. So the whole family, all the men in the family are colorblind, which is interesting. But that website shows you what it would look like if you had different kinds of color blindness. So it will show you an image, say a scene of a market, and then there'll be a filter that would show you, okay, well, what if you were blue insensitive? Or what if you were green insensitive? And you can kind of flip through and see how different scenes would look for different kinds of color blindness. I just find it an interesting way to actually see it in action, because for those of us who see colors, it's kind of hard to imagine how you might see it differently. So just a neat thing if you wanted to spend some time on it. And so as we said, the trichromatic theory does a good job of some things. It matches the fact that we have three different types of cones when we look at our eye structures. But it doesn't do a fantastic job with yellow, and there are some other things that it can't explain. So we have a second theory that we can talk about. And this is the opponent process theory. So this one was proposed by Edward Heering, and this is still in the 1800s, sort of a little bit later on in the 1800s, but he was really upset that the standing theory didn't do a great job with yellow. So he said, okay, let's propose something else that does a good job explaining yellow and maybe some of the other stuff that the trichromatic theory can't explain. So let's look at what the theory says, and you're going to find it starts off very similar. So the human eye has 3 types of receptors that are sensitive to different wavelengths of light. That's exactly the same as the other one. But where it starts to change is what it's sensitive to and how. So in this case, for the opponent process theory, they're going with a different idea. So instead of having sort of one set of cones that does red, one that does green, and one that does blue, they have 3 sets that respond to pairs of wavelengths. So they have a cone that does red or green. So it would send one signal if it's experiencing red, and a different signal if it's experiencing green. Without getting into sort of the specifics of it, think of it as when they're sending a signal, it's saying that it's red, and when that signal is turned off, it's saying that it's green. So it has sort of a switch where it'll say we either have red or green. And then if we look at our spectrum here, the reds and the greens are fairly separate from one another. So the reds are all the way to that side and the greens are in the middle. So you shouldn't be seeing both in the exact same place because then they would be mixing. So it makes sense that those would be separated and that we could have a signal for red or green. So that's one of our types of cones. Our second type of cone would send a signal for either blue or yellow. And so this is where we introduce that sensitivity to yellow. And again, if we scroll back, our blue is towards the far left, and then the yellow is over in the middle between the green and the red. So blue and yellow aren't adjacent to one another, so they shouldn't touch. So we could have a signal that says we either have blue or we have yellow. And then the last of our types here is just going to give us a black or white. So kind of similar to what our rods do. So presence of light or absence of light. And the reason why this theory was proposed and why it stuck around for quite a while is because it, A, explains yellow, which was a problem, and B, it does a great job explaining something called after images. And so if you've taken high school or even grade school in Canada and you've done sort of science or social studies, they'll show you this image and they'll get you to stare at that black dot in the middle and they say, don't blink, don't move your eyes, stare at this for 20 seconds and then stare at a blank piece of paper. And what should happen is when you look away from that dot after staring at it and you look at a white wall or a white piece of paper, then we'll see a reversal of those colors. So the green will start looking red and the black will start looking white, which ends up making sense because green is paired with red. So when our eyes are tired of seeing green, when we've stared at it for a long time and tired that cell out, when we look away, it's going to overcompensate and show red instead. And then for black, same thing. It looked at black for too long, it got tired out, it's going to overcompensate when we look away, and we see white. So these after images can absolutely be explained by this opponent process theory, but they cannot be touched by the trichromatic theory. There's no explanation for them in that theory. So that's why this one does a good job of other things. And so the conclusion that we end up drawing here is that they both do a good job explaining some things, but not others. So what's the real case? What's actually going on? And they've done kind of the simplest thing here, and the current theory in its simplest form is a straight combination of both the trichromatic and opponent process theories. So we go back to that, yes, there are 3 types of cones that are sensitive to 3 different kinds of wavelengths. But if we scroll to that next slide, we're starting at the top, those are our cones, and we've returned to the blue, green, and red. So we're using that from the trichromatic theory. But instead of just having 3 types of cones, we're now talking about another type of cell that will act like the opponent process theory proposed. Wrong way. So these are our lateral geniculate nucleus cells, or LGN cells, because that is way too hard to say repeatedly. And so those would be cells in both the retina and or the visual cortex, and they do that opponent processing. So they would respond to either blue or yellow, red or green, black or white. And so we would add in that extra row there where the signal from our blue cones would feed into an opponent cell, what we now know to be our LGN cells. And so we'll get a little bit of a signal from the blue there, and we'd also get a combination of the red and the green to feed that yellow side of that cell. So those cells do processing to tell us, are we seeing blue or yellow? Are we seeing green or red? Are we seeing black or white? So we have both our three kinds of cones and three kinds of opponent cells, but they're not the same thing, they're just sort of slightly different. So this is our, I don't even know if I said it, this is our dual process theory that involves both of those processes just sort of combined together in a way that seems to work. And again, we can go into tons more detail on this, but there's an entire class on that, so we just leave it at this point. So the next thing that we want to touch on fairly briefly is the concept of, okay, so this is the science behind what we're seeing, but how do we turn that into meaning? How do we get perception out of it? So we've been building up from sort of the biological basis, and then we get into how do we combine things, and now we're into, okay, well, how do we get meaning or extra information from that? So we're going to look at depth perception, and then we're going to get into some of our other ways of organizing information that we see. So for depth perception, this is when we interpret visual cues to give us some indication of how close or far away something is. So at the front of the room here, you can see that my microphone is closer to you than I am. And there are a bunch of different cues that can tell you that. And so we can look at either monocular or binocular cues. And what these are doing is, again, we can look at the roots there. So mono meaning one, bi meaning two, and ocular meaning eyes. So monocular cues only need one functioning eye to work. Binocular cues need two. And I always find this really interesting because my father doesn't have sight in one of his eyes. So he taught me to play baseball and everything, but none of his tips or tricks on, playing baseball and catching a ball properly actually involved binocular cues because he doesn't have any. So I've been hit in the face multiple times with balls. So one of the downsides of trying to play sports when you lack that kind of depth perception. But we can talk very, very briefly about what we can see here. So the monocular cues, I find the best way to frame these is if you've ever taken an art class or if you've ever looked at a painting, something that's two-dimensional but gives the impression of being three-dimensional, it's using monocular cues. Almost all of those 2D things are making use of things you don't need to see with both eyes. So occlusion, that would be if you're sitting somewhere where my microphone is between you and me. So if the microphone is occluding me, if it's blocking me from your view, then it must be closer to you. We can look at relative height. So we know that people tend to be about human height. So if you see someone closer to you or further away, they will look a little bit taller or shorter. And there's a lot of variation in there, but we can get a decent judgment of how large something might be. Same thing with relative size. So if we're looking at a car and it looks really small in our vision or really close or really big in our vision, we can tell if it's close or far away. We also have something really neat called perspective convergence. So if you ever had a teacher who got you to draw a perspective horizon, where you draw sort of a horizon point and you do that vanishing point where you connect usually like railroad tracks or the sides of a street, or if you were looking at power lines or something like that, and as they get further away they get smaller and they fade into the distance. That's using perspective convergence, where everything sort of condenses down to a single point. And then we can rely on familiar size, which is kind of like that relative height and relative size, where instead of just relying on, okay, well, there's two cars and one is much larger than the other, so the larger one must be closer, we can just say, well, we know how big cars tend to be, so I can guesstimate how far away it is based on what I know. And then the last one, atmospheric perspective, is kind of neat because as things get further away, there's more air between us and it. So as it gets further away, it gets a little bit harder to see. And you find it a lot more noticeable, say, this summer when we had all of those smoke days. So it's not just air, now we have haze in between us. And so things that were further away look a lot less colorful, so there's duller, and they get a little bit fuzzy, they're not as clear or sharp, and so you get a lot less detail in them as well. So that difference in sort of the saturation of their color and the amount of detail and the clarity will change whether they're close or far away. And that even works, so if we think about it the other way around, so if it's closer to you, things that are up close, you can see with a lot more detail. They tend to be a lot brighter and more colorful, in addition to being larger. So these are all different kinds of monocular depth cues, and we can have a whole bunch of different ones of these. Question?
Ma'am, what is the basic difference between occlusion and perspective convergence?
So occlusion is where one thing is blocking another thing. So the thing that is closer to you can occlude or cover up the thing that's behind it. So you can't see my legs because the stand here is in the way. So this is occlusion. You know that I'm further away because you can see the stand but not the rest of me. Whereas if I move and I stand in front of it, then I'm occluding the podium, and so I must be closer. And then perspective convergence is just when, actually, they do have markers. This might be a little bit easier to visualize, but it's where they get you to draw sort of that perspective line, and you would draw like a highway or whatever with your power lines, and it would look something sort of like that, where things fade and get smaller into the distance. So that would be perspective convergence because the lines are converging or coming together as they get further away. Does that kind of help?
What lines? Like which kind of lines?
Any kind of lines. Lines in the environment. So this is something that the road is not getting closer together. A road is always going to be however wide it is. So it's 2 lanes wide. And so it should run parallel. But as it gets further away from you, it looks like it's getting closer. So it's an illusion that lets us perceive distance. Awesome. All right. So those are sort of those visual tricks that help us see distance. Our binocular cues actually come from the quality of us having 2 eyes that face forward. So we can rely on the fact that we have 2 slightly differently placed eyes, and we can use information and differences between the 2 of them to tell us how near or far something is. So this is kind of similar to using our opposite ears to tell where a sound is. So you can localize sound based on ear placement. Same thing, but with eyes. And so there's 2 big ones that we can look at here. The first is retinal disparity. And that's that idea where, actually the easiest way to do it is if you put your finger in front of your eye and you sort of close one eye and then the other. And you can see that your finger seems to shuffle just a little bit back and forth. That happens because one of our eyes tends to be dominant over the other, but also because they're looking at something from a slightly different perspective. And as things are getting closer and closer to you, we see a bigger jump in their position because this eye is seeing it sort of on this side, and this eye is seeing it on that side. And we can look at that difference, that disparity, and tell how far away something is. And you don't have to sort of close one eye and then the other, but that's the easiest way to notice it. When we're looking at things in the world, we're just sort of passively aware of that. So they see slightly different views, and our brains can interpret the amount of difference as sort of how far or close something is. And the next one is convergence, and this is an interesting one where you can actually, again, not that you're consciously aware of it, but you notice how much your eyes are bending inward to look at something. So you've noticed if you sort of look at your finger out there and then bring it closer, your eyes sort of bend inward. We have that convergence again, kind of like the lines looking like they're coming together, but this time it's your pupils coming together. And as things are further away, your eyes are going to sort of spread outward and look parallel to one another. And so our brain can pick up on how much is our sort of line of sight bending in or out to tell us how far the thing is that we're focusing on. Okay, so those are our different kinds of depth cues. And then next is actually one of my favorite parts of this chapter, and even maybe some of all of the chapters. We have to keep it a little bit short because we could spend forever talking about optical illusions, but I do want to get into a little bit of how we process things and briefly mention how once you know how we process things, how we treat information, how we put things together, we can kind of trick our brains into seeing things that aren't quite there. So that's how most optical illusions work. They're going to use typically monocular cues that mislead us into interpreting an image a way that it shouldn't be interpreted. But first we're going to talk about our two forms of processing. And this is bottom-up versus top-down processing. There are tons of different ways that we can frame this, but with our bottom-up processing, we're emphasizing sort of the individual details or the individual components of a particular image. So you're going to look at those individual stimuli, those little pieces that come together to give you that whole. So start with the individual elements and start putting them together in order to interpret the object as a whole. So on our image here, they start with detecting just basic features. What color is it? What shape is it? What size? Those sorts of things, and start putting it together. Okay, well, we've seen sort of a vertical line and a horizontal line, and then another vertical line and a horizontal line. It's a square. What does that mean? What does that limit us to? And then we can start sort of putting together all of those features to get some kind of recognition. So maybe it's a box, and we've sort of put it together from all of the individual lines that make up that box. The other hand, we have top-down processing, and this is where we start interpreting our sensory information. Oops. Oh, please don't scroll. There we go. Okay. We interpret our sensory information based on what we already know. So as we get older and as we have more experience with our environment, we know more things. So when we see stuff, we don't have to figure out exactly what it is every single time. And so the example that we can use is looking at an exit sign. Every time you see an exit sign, you don't necessarily go, okay, well, there's sort of the downward line and then the 3 sideways lines for the E, and then there's 2 cross lines for the X, and go on from there. We know that those red signs that tend to be in all buildings, that's an exit sign. So we have our pre-existing knowledge. We can take one look at it and go, oh yeah, we know exactly what it is based on context and experience. So we can form a perceptual hypothesis. So when we see it, yeah, it's probably an exit sign. We can look closer at it to make sure. Does it actually say exit or does it say something else? But we can eventually have our recognition after we've sort of double-checked our gut response at first. But that top-down processing lets us do stuff more quickly and maybe not using as many resources as we would if we had to figure it out piece by piece. And so we have a couple of examples here where the one on the left, is it a duck or is it a rabbit? That's one of those ones that causes problems because we can do the bottom-up processing. And we can see, yeah, there's an eye, but are those ears or is that a beak? Is it a duck or is it a rabbit? And how we put it together will give us a different perspective overall. And then on the other side, the exit sign is the example we just used of the top-down processing. And so we use a combination of both of them, and they can both have sort of their pros and cons depending on the situation. One of the interesting ones that I heard, I don't think it's from our textbook, but it's from one of the ones I've used over the years. They talked about someone who had a stall, or actually a small store at a farmer's market, and they sold beads, so you could make jewelry. But they were surrounded by food stalls. And so people came in regularly thinking it was a bread store. 1 letter difference. but based on context and location and starts with a B and ends with a D, they assumed bread, not beads. And so they had lots of issues with that and had to sort of rethink their signage. But that would be one of those ways that top-down processing can kind of throw us for a loop. All right, our next couple of topics are going to be a very quick overview of Gestalt psychology. And so Gestalt, as we care about him, was very, very focused on how we perceive forms. So he had this idea that with humans, we tend to perceive the whole as being a lot more than each of the individual components. Humans are really good at putting things together, finding relationships and picking out information from our environment. And so he wanted to have different rules, different principles that would describe how these get put together. So the first of these, the easiest one for me at least, is the figure-ground relationship. And so if I hop to that next slide, this basically says that people are really good at identifying what is the figure, what is the focal point, what's the attention place, and what is the background. So if we look at the stuff at the bottom, those are optical illusions that actually play with our ability to decide on what's the focal point. So we can't decide, is it, so the one in the middle, is it a chalice or is it two faces? And if we can't decide which one is that figure and which one is the background, our mind kind of jumps back and forth between the two. On the sort of far right there, we have, is it a face or is it a landscape with a person walking through it? And then this one here, this is the one that always drives me nuts. It's, is it a young woman looking away or is it an old woman with sort of a headscarf? And I can never for the life of me see the old woman in that image. And I have taught this course for many, many years, and I'm told that it's there. But maybe you can, or maybe you cannot see it. But this is sort of making use of that issue where we're usually good at categorizing figures and focal points versus backgrounds, and we can actually make sort of tricks of that. The next of our principles are all about, sort of the four in the middle are all about how we organize items that are sort of nearby or similar or that should have some kind of pattern to them. And I'm going to hop to another slide in just a second and show all of those individually, but I also wanted to mention the last one, which is the principle of common fate. And this is the idea that when things are moving and acting together, we tend to assume that they are associated with one another. So the best example of this is if you look at a giant flock of starlings. That would be called a murmuration in case you're curious. But they tend to move all together. They move as one, and they create weird shapes in the sky. But when we look at it, we don't tend to see a whole bunch of individual birds. We tend to see the shape of the flock as a whole. So that principle of common fate has told us that because they're in the same place and they're moving together, treat them as one. So we don't focus on each individual one, we treat them as a group. And that's going to be pretty consistent through our other principles. Good, and we're still doing great on time today. So for our principle of similarity, when we look at all of these circles here, we tend to see two triangles that we group based on how similar they are to each other. So an inner triangle of small circles and an outer triangle with three large circles. Or maybe we see an inner triangle of small circles and then three separate large circles. But that's going to be how we group things. So things that are very similar to one another tend to be treated the same. So we assume that it's a triangle of small circles and not sort of random assortments of circles there. For proximity, here we add space in there. And so instead of treating all the small lines as the same and all the large lines as the same, because of that space, we have three pairs. So a small and a large line are together. They're in close proximity to each other, and we tend to associate them together. So we would view this as three groups of two instead of looking at three small lines and three large lines. So that proximity, that closeness, that location matters to us. Our principle of closure is just our sort of baseline assumption that things are supposed to be complete. And this comes from the fact that in nature, if something isn't completely a full shape, it's usually that principle of occlusion, where something is hidden behind something else. So maybe that partial circle, you can't see that one side because maybe there's a block in front of it. So we can only see, say, 80% of the circle because something's in the way. So our brain fills in what's missing and says, oh, it's definitely a circle, we just can't see it all. So we assume that it should be filled in, and we can continue the pattern to connect it. And that sort of continuing a pattern also comes in with our principle of continuity. And so for this one, we tend to assume patterns, that things continue. So if we look at those lines of dots, we would probably see it as a line that goes from A to B, and another line that goes from C to D, as opposed to, say, two kind of shark fin shapes that go from A to the middle to C, and B to the middle to D. So we would probably see two continuous lines as opposed to those separate shark fin shapes. And so these are just ways of describing how we tend to treat information. We also can talk about things like perceptual constancies. And this kind of goes back to that idea of knowing that things tend to be certain sizes, and we can use our knowledge of relative sizes to tell us how close or far something is. This is a really cool way of looking at that. So if we look at size constancy and go back to that idea that people tend to be about human-sized, when we look at that first image on the left, we don't assume that it is one large person and one small person. We assume that it's two people and one is just sitting further back. And then if we move the person from the background up closer, so we actually see how different in size they really are, it seems like that's way too extreme. He's way too small in that second picture. But if you went and measured it, it's exactly the same. It was just photoshopped to a different position. So the person in the right image, that small person, looks much, much smaller than the person on the left because of our understanding of size constancies. So we kind of account for distance when we're looking at things without even being aware of it. And we do the same thing for shape. So if you're having a conversation with a friend and they need to move to the other side of you, so they go from over here to over there, well, the shape of their face might be a little bit different. It's going to be the opposite. But you know it's still your friend. It's still the same person. You're just looking at them from a slightly different angle. The shape is the exact same, but your perspective has changed. Same with brightness. So when you're inside versus outside, your shirt might look like a different color. So if you're under fluorescent light versus sunlight versus candlelight, it's going to look very different, but we know that it's because it's being lit differently, not because you have a magical color-changing shirt. And so same with hue as well, so we can have different colors, that same thing where brightness can change, our perception of color can change based on what kind of light is shining on us. So if our light is sort of warm and yellowy like candlelight, it can change the color that it looks to be. And then also location in space, that's kind of like that retinal disparity, where something can look like it's on this side if you're using this eye, and looks on that side if you're using that eye. As we move around things, it looks like it's changing position, it looks like it's moving, but in fact it's us moving. Everything else tends to stay in one place. And so these are all things that our brain is capable of sort of recognizing and sort of accounting for when we're looking at stuff. But after having this lecture, I always find it kind of weird to walk around and think, okay, somehow my brain figured out that as I walk towards the back of the classroom, the chairs aren't moving towards me, they're still, and I'm moving towards them. Or looking at sort of driving in a slightly different way, and things like that. But there's lots and lots of stuff that our brain can do in the background, and these are just a couple of the highlights of it. But yeah, so I will leave it there for today. I think we are actually just a little bit early today in terms of timing, which is really nice. But we will start